A very important and unique feature of the metabolism of nucleotides is that they are not only they are formed from the scratch, I mean, where different components from are gathered from the raw materials and they are formed, but also they are salvaged. That is, they are made from already made components, already made nitrogenous bases, and only what you need is to, under the action of certain enzyme, one or two reaction, and you end up with the formation of the corresponding nucleosides. So the nucleosides which we form from, for instance, the diet or from the degradation of the nucleic acid, they can be these whole components, like let's say if purine compounds like adenine or hypoxanthine or guanine, you get from your diet or from the nucleotides, or sorry, from the nucleic acid, that can be then salvaged for the formation of corresponding nucleosides like inosine monophosphate, like guanosine monophosphate, like adenine monophosphate. So one of the use of this salvage process is that you can get the formation of these, you can get these nucleosides with the expenditure of minimized energy. And this is very important for the tissues or for the organs where energy is very much needed, where this saving the energy is of paramount importance. For instance, in your brain, I mean, now whenever I'm talking, my brain is using a lot of energy, and when you are listening to me, your brain is using a lot of energy on thinking process. What I have to talk, I have to think. What you listen, you have to think and ponder. And this needs a lot of energy. So here in brain, then, rather than utilizing the whole energy or most of the energy in formation of the compounds from the scratch, it's much easier if you salvage the formation of these compounds. So here, this is a very important feature as far as the synthesis of the nucleosides is concerned. The two enzymes are HGPRT and APRT for adenine phosphoribosyl transferase, and HGPRT, hypoxanthine guanine phosphoribosyl transferase. These enzymes are important in the formation of these nucleosides through the salvage process.